US and Europe. So around 50% of acute liver failure in Western population is because of drug overdose, especially paracetamol. Now, other important thing which we need to consider is that Delhi is responsible for it's a major reason for discontinuation of drugs, because during preclinical or clinical trials, you will find that a lot of these have adverse events in the form of liver injury and they get discontinued. So this is a data from 1975 to 2007, in which 32% of drug withdrawals were attributed to hepatotoxicity. Now, these pie charts, I'd like to draw your attention towards those red areas. So different drugs from 2000 to 2010. So these red areas show the presence of liver toxicity in 100% of people. So it ranges from 2 to 10%. And some important group of drugs like troglitazone got discontinued because of presence of So one is latency. So from the onset of intake to either abnormal liver function test or to development of symptoms. D challenge is the symptoms resolve, the time taken for symptoms to resolve. Re-challenge, recurrence of symptoms on restarting that culprit agent. Likelihood means the potency or the we are aware of likelihood of that agent being hepatotoxic, and phenotype is the clinical manifestation. So these are different terms which you use in defining Delhi. Now, epidemiologically, the data is varied. We have majority of data from West, but to begin with from Asia. So this was a large study which was conducted in Korea, and the incidence of hospitalization was around 12 cases per 100,000 population. And this has been now shown that the prevalence of the incidence of drug-induced liver injury because of herbal and traditional medications is rising in Asian population. So it's increased from 7% to 11% in West, but in Asian countries, this was the major database which was published in 2004 from General Practice Research database in UK. And they found the incidence to be about 2.4 cases per 100,000 population, and similar data of 2.7 cases per 100,000 population in USA. There, the majority of drug-induced liver injury is because of antibiotics, of which augmentin is or amoxiclav is the most common culprit agent. So what we need to differentiate that paracetamol is the most common cause of acute liver failure in Western population, but amoxiclav is the most common culprit agent for drug-induced liver injury. Now we have some interesting data which has come from India, which got published in 2021 in Journal of Clinical and Experimental Hepatology. This was a 5-year period data collection from 2013 to 18, and it included 1288 patients across more than 40 centers throughout India. Now, what this data shows that 99% of drug-induced liver injury in India is idiosyncratic. 50%, so equal distribution of males and females. 68% of these patients had jaundice. We know that HIV drugs are hepatotoxic. Skin reactions, 19%, ascites in 16%, and hepatic encephalopathy happened in 10%. So presence of increased INR and hepatic encephalopathy means that the disease is severe, which we'll talk further. 32.4% of patients had severe disease. That is, the INR was more than 1.5, or they had developed ascites or hepatic encephalopathy. So out of 1288, we are talking of around 150 patients who actually succumbed to this injury. Now, of these subgroup analysis, 65% of these patients had hepatic encephalopathy. 6% of patients fulfilled Hy's law, which you all are aware of. What is that? We'll talk in further slides. and 16% of these had jaundice. So this is all we all know. So the most common culprit in India still is anti-tubercular drugs, which was there in around 50% of patients. Traditional and alternative medicines are now increasing, and we have 14%. First generation anti-epileptics like phenytoin, carbamazepine, we do not see paracetamol-induced acute liver failure. The majority of our acute liver failures because of Delhi are because of anti-tubercular therapy. So what, just to add to our Indian data, so last couple of years in COVID, we had these a lot of drugs being used by patients as immune boosters. And in India, North India, we call it as Giloy. It is also called as Stinospora cordifolia. And there have been initially this case series came from Mumbai group in 2020. And subsequently, this was a multicentric Indian data of, excuse me, of 43 patients of Giloy-induced liver injury. So for our Indian population, Ayurvedic medications are considered, they have a reputation of having less side effects. But now the data is emerging that they may not be as safe as we consider them. So what happened in these group of patients when they were biopsied, what biopsy showed that there was some said is that it is associated with autoimmune, with acute hepatitis with autoimmune-like features. Now, we all know, we are sitting in a pharmacology community, that classically Delhi has been characterized as two types, direct and idiosyncratic. Now, there is a third group which is now getting added, and which is called as indirect hepatotoxicity. So what do we mean by direct? So it is common, it is dose dependent, and it is predictable. So like acetaminophen. If you increase the dose of acetaminophen, if it's more than 7.5 grams, the person lands up in acute hepatitis, subgroup goes into acute liver failure. In comparison, idiosyncratic drug reactions are rare. They are not dose dependent. They are not predictable and cannot be replicated in animal models. So what causes direct hepatotoxicity? It is the intrinsic hepatotoxic nature of the molecule. So the metabolite which gets produced causes damage to hepatocyte. Idiosyncratic reactions are idiosyncratic. Injury. Now, what is indirect hepatotoxicity? So this is a new group which has been added. So there will be some group of drugs which will not be directly causing liver injury, but by immunosuppression, they can cause activation of hepatitis, which can damage liver, or there could be a subgroup of patients where there can be fatty accumulation in the liver, like steroids, anabolic steroids, amiodarone. All these have been now considered as part of indirect hepatotoxicity, which is a new classification which has been recently added. So we all know about drug metabolism. When the drug gets ingested, it goes through phase 1 and phase 2 reactions. And so let's take an example of alcohol. So once the person consumes alcohol, it gets converted by alcohol dehydrogenase into acetaldehyde. Now, acetaldehyde is a part of phase 1 reaction. And when we know that acetaldehyde immediately gets converted into acetate and it gets excreted. But what causes injury in alcoholics? It is the acetaldehyde. So this is one prototype which I would share with you in terms of explaining this process. The metabolism should be of drug should be lipophilic and it should be dose. So indirect hepatotoxicity. Now through phase 1, it leads to formation of reactive metabolites. So in alcohol, let's take it as acetaldehyde. Now, what happens in hepatocytes? They are protective mechanisms. If they fail, like the glutathione gets depleted. So we know that glutathione is a free radical scavenger. So these metabolites which get formed, they can get covalent binding with the protein and they can form adducts which are known as haptens. Then it can lead to mitochondrial stress, leading to reactive oxygen species release and endoplasmic reticular stress. So in the direct hepatotoxicity, intrinsic DILI, what happens is the amount of dose becomes the determining factor for liver injury. In idiosyncratic, so these adducts act as neoantigens or haptens and they stimulate immune system. They get expressed on hepatocyte surface, it stimulates immune system, and then there is an immune mediated injury in a person who has genetic predisposition. Now, we cannot have any gastroenterology like and it is an interesting topic where now there is a data to say that the effect of anti-tumor drugs depends on the gut microbiome of individual. And I could come across this interesting article, which experiment which took place in mice, where they have shown that there is a diurnal variation of paracetamol hepatotoxicity. The details are a bit complicated for me to share, and it was difficult for me to understand because it was at a very poor pharmacokinetic level. So kindly excuse me for that. So what are the risk factors? Age. So more the age, like Dr. Meenakshi is head of geriatrics, more the age, it increases the risk of drug-induced liver injury. Gender, we'll talk about it. Pregnancy, the data is not much there because majority of drugs are not used in pregnancy because of their teratogenic, or the gynecologists are afraid and they in using those drugs. What happens with alcohol, underlying liver disease, and genetics? So we'll briefly talk. And drug-related factors are dose, metabolism, and lipophilicity. So what happens is with the advancing in age, if they get DILI, they tend to progress more to acute liver failure. As I mentioned, pregnancy, we have limited data. So what happens is alcohol intake stimulates enzymes in liver. So it stimulates cytochrome P450 system, and it has been shown to have more injury with isoniazid, methotrexate, and halothane. We know that underlying chronic liver disease. So a patient with cirrhosis diagnosed to have tuberculosis, our guidelines say, if you are starting ATT, please monitor these patients with frequent LFTs because there is an increased risk of DILI in these patients. So this risk is around 3 times. And a benign condition like non-alcoholic fatty liver disease, which is part of chronic liver disease, also increases the risk of DILI. And there are different single nucleotide polymorphisms in numerous genes and HLA regions, which have been shown to have some impact on DILI. Coming to drug-related factors. So drugs which have doses which exceed more than 50 milligram tend to have higher incidence of DILI. Oral medications who have that is, their lipophilicity is more than 3. And if their daily dosing is more than 100 milligram, then these drugs have increased risk of DILI. So how do you define DILI? So majority of these patients will have biochemical DILI. Symptomatic DILI will be less frequent. Majority of patients will have clinical biochemical DILI. So your liver enzymes, if it gets elevated more than 5 times, alkaline phosphatase is a marker of cholestasis, rises more than 2 times, or total bilirubin increases more than 2-fold, but enzymes are more than 3. So there is another ratio based on liver function test, which you call as R ratio. So what R ratio does is, it defines aminotransferases ratio in comparison to alkaline phosphatase. So suppose in my lab, the upper limit of ALT is 40 and the person has ALT value of 400. So he has 10 times upper limit of normal. His alkaline phosphatase, my upper limit of normal is 50. His alkaline phosphatase is 75. So it is 1.5 times. So if I calculate the ratio of ALT to ALP, so it becomes more than 5. If it is more than 5, it indicates cholestatic. Less than 2 indicates more than 5 indicates hepatocellular. Less than 2 indicates cholestatic, and between is mixed injury. Like US Delhi Network and International Delhi Expert Working Group. If you look at this, what you will find is that mild, moderate, severe is based on only LFT parameters. What makes us severe is if your INR is more than 1.5, if the person develops hepatic encephalopathy, or if the patient has ascites. So these are poor prognostic indicators and it indicate that the person has severe DILI. If I have a study protocol where I say, let's collect data from AIIMS Rishikesh and I invite gynecology, pharmacology, surgery, and all other departments to send me patients. So how will I assess that the drug is implicated in that particular hepatotoxicity? We do it by a causality assessment tool, which is called as RUCAM. This is Russell-Uclaf causality assessment method, which comprises of 5 parameters, that is latency, D-challenge, risk factors, co-medications, exclusion of other etiologies, data on drug hepatotoxicity, and re-challenge. So it creates a score, and if it's more than 8, it is highly likely that the person is having drug-induced liver injury. Industry and clinicians. It has poor interrelated reliability, and it has some issues with arbitrary scoring. So clinical manifestations, as I said, some patients will be asymptomatic, some patients will become symptomatic. So like a person who has hepatitic-like illness, so these patients have nausea, vomiting, some of these patients will end up in jaundice. Hypersensitivity reactions are seen in less than 1/3 patients. So you do not find many of these patients will have rash, eosinophilia, joint pains, petechiae. It happens in less than 1/3 of patients. Okay. So once you have a person who has presented as an acute liver injury, unexplained chronic hepatitis, unexplained worsening of CLD or ACLF, suspect DILI. So ask history. So yesterday we admitted one patient from our OPD. He was a case of acute on chronic liver failure. So his chronic liver disease was ethanol. But now the patient had been abstinent for alcohol for 2 years. So this becomes a suspect for drug-induced liver injury. That is a different issue, that there is some confusion. Wife says that he still consumes alcohol, but he categorically denies. So it is a diagnosis of exclusion. So you need to rule out other causes like viral hepatitis, autoimmune hepatitis, vascular abnormalities. So once you have labeled it as DILI, so the important thing in these group of patients will be, when we'll talk of treatment, which will be very brief because there is no much specific treatment. So it is a diagnosis of exclusion, and you need to rule out other important etiologies of liver disease. Briefly discuss about new biomarkers. So traditional biomarker for DILI is LFT. Apart from that, you do not have. For paracetamol poisoning, you measure some metabolite, but majority of patients you do not get. So there is some active research which is going on. So for early stage detection, you have microscopic RNAs, glutamate dehydrogenase, cytokeratin molecules, and all those which are in the pipeline, but it has not been still in the clinical practice. So the most the Ramban is an acetylcysteine. For valproate, you need carnitine. Then there is something which is coming up new in a big way. A lot of pharmacology people will be aware of this. That is immune checkpoint inhibitors. Immune checkpoint inhibitors are the new class of molecules which are anti-tumor molecules, and they have been shown to associate with liver toxicity. For them, the treatment of choice is steroids. So I like to conclude by saying that DILI is uncommon. So you need a high index of suspicion. It leads to significant hepatic morbidity and mortality, and newer biomarkers for early diagnosis are the need of the hour. Thank you.
Thank you, Dr. Rohit, for this wonderful talk. You really enlightened us, and we will become more sensitive towards this liver injury due to the drugs, and especially me because I'm dealing with the geriatrics. So the now house is open for the discussion. Any questions from the audience?
Dr. Rohit, I wanted to ask, like in your presentation, you said the Ayurvedic medicines are the second common cause of drug-induced liver injury. So we have to confirm whether it is because of drug and Ayurvedic medicine, because many of the Indian patients, they take allopathic as well as Ayurvedic medicines together.
Okay, thank you for this question, ma'am. So the problem with a lot of these herbal and traditional medications is that they do not come as a single prescription. So these are polypharmacy. So a syrup of some of these preparations, if you go through their index or labeling, that you will find that it comprises of 10 different classes of drugs. So it becomes extremely difficult for you to actually pinpoint, and this has been highlighted in literature, not only in India, but from the West also, that it is extremely difficult to pinpoint that of all those combinations, which one is responsible. So a lot of these patients, ma'am, those who do not have history of concomitant so-called allopathic treatment, so like this Giloy patients, which has been seen during COVID pandemic, these patients were not on any other drugs. These were predominantly on immune booster in the form of Giloy. So your point is very valid that it would be extremely difficult to actually synthesize that what is contributing to what. But in those patients where there is no concomitant known hepatotoxic drugs, you label them as because of herbal and traditional medicines, but it's difficult in those lists which one is hepatotoxic.
Exactly. And many anti-epileptic Ayurvedic medicines, they have been shown in literature to contain steroids also, right?
Contain?
Many steroids, corticosteroids. Yeah.
So our Rishikesh is famous for that. You all must be aware of an individual who was there, who had been treating anti-epileptic patients, and then in those early days, if some of you must be young, you could see his picture coming up with a lot of celebrities, political celebrities at that time. And when ultimately uska chhapa pada and people found out that instead of using herbal and traditional medicines, he was using first-line anti-epileptics along with steroids. So that is there.
Yes. So, Dr. Rohit, I just want to ask one question. Do you think that there is some role of so-called hepatoprotective drugs? So when we are prescribing a medicine and we know it is hepatotoxic, but still we have to give it. So is there any, for example,
protective drugs which are frequently used by general practitioners, one is LIV52. It's an Ayurvedic preparation, and another is ursodeoxycholic acid. So a study which was conducted from PGI Chandigarh, I think in 2003-4, I've forgotten the exact date. So it found that LIV52 acts as a placebo. You use it, you do not use it, it doesn't help you. As far as hepatoprotective drugs are concerned, for all practical purposes, there is no hepatoprotective drug available across the world. I think we give for our own satisfaction rather than any clinical benefit these drugs have.
Thank you. Thank you, Dr. Rohit.
Thank you.
Any other question? Otherwise, yes.
Thank you, Dr. Rohit, for a wonderful lecture. Seroglitazar.
Saroglitazar. Okay. So, saroglitazar is a nucleate in the block. It's an Indian product introduced by Zydus Pharma. So it's a PPAR alpha gamma agonist, which has been, so it was produced, so its initial indication was diabetic dyslipidemia. Now it has been approved by DGCI for treatment of non-alcoholic fatty liver disease. So currently there are no indications for use of saroglitazar for prevention of a treatment of drug-induced liver injury.
Thank you, sir, for your wonderful presentation, sir. Sir, I have a query. What are the most common traditional and alternative medicine which causes liver injury in Indian population, sir?
In
Indian population.
Indian population?
Yes, sir.
Okay. So,
Sir, basically, I work on traditional, means plant extract in experiment.
products, I'm sorry for that, because these are now broadly classified as herbal and traditional medicines.
Yes.
So I could come across the Stanospora or Giloy as one of the latest to be included. Rest will be speculative on that part. I'm sorry for not answering your question.
Thank you, sir.
See you.
It is an informative lecture. Silymarin is one of the widely used standard hepatoprotective agent in many of the research studies. Is it having any role in the clinical practice as a hepatoprotective agent?
So if you, the slides are not being projected. So what we need to understand is that there is, as a gastroenterologist, I can say that there is for protection of liver. None of these work. Frankly, none of these work. Like Live 52, because it's the most popular brand, I'm using it here. But none of these actually work. If you do a trial, you don't find much difference in using this or using a placebo. So one indication for silymarin is mushroom poisoning. So there is a subgroup of mushroom which is found in Uttarakhand, also in Himalayas. Around last year, we had 3 patients from one family who landed up in acute liver failure. Unfortunately, they all succumbed to illness. There is some role of penicillin G and silymarin in that group of patients, but generally using it as a hepatoprotective agent doesn't work. Thank you.
Thank you, sir. Thank you, sir.
If there are no questions, we thank the speaker, Dr. Rohit Gupta, for the wonderful presentation.
Thank you. Thank you for this option.
I think we can now invite the second speaker, a very dynamic pharmacovigilance professional, Ms. Arshia Bhandari. Is he present here?
Madam, may I ask one question? Hello, madam, can I ask one question to sir?
Yes, please.
Okay, so thank
To
you,
the
madam. Thank you, madam, for the opportunity. Because sir was mentioning about the NFLD or MFLD, whatever now today it is being called, the metabolic syndrome associated fatty liver disease. And often we came across a little bit of elevated SGPT, SGOT in our diabetic subjects. So in those group, when we are initiating statin, there is some fear in our mind. It could also lead to some hepatitis or statin-induced hepatitis could be there. So how should we approach and how should we be vigilant while initiating statin in this subset?
Thank you for your question. It's an excellent question. So his question is, if I can repeat for the sake of audience, is hepatotoxic potential of statins. So when statins got introduced patients with mild deranged liver function, and then there is a concept called as adaptation, which you pharmacologists are very well aware of, that with a period of continuous usage of statins, your LFTs tend to settle down. So there is also, answering to your question, that there is not enough data of usage of statins in NAFLD with deranged liver enzymes, where it has proven to be safe and it helps in control of hypertriglyceridemia and it decreases liver fat. So the only thing is that once you start statins, get a LFT done. If you are fulfilling all those criteria of severe liver injury, then kindly discontinue. Otherwise, mild derangement of liver function test at the onset or on drugs, you can continue statins safely. And there is a phenomena which is called as adaptation.
Thank you.
Thank you, sir. So, sir, we can take the cutoff as 3 times upper limit of normal, or any specific cutoff you want to mention?
So the cutoff from my side will be 5 times upper limit of normal if you're talking of enzyme.
Okay, sir. Thank you, sir.
Second speaker, Ms. Arshia Bandari, who is a pharmacovigilance professional with more than 16 years of experience. She is the founder of PHV Fit, and she has published many publications, which has been published in the Uppsala reports. And she is a qualified medication safety specialist and a reviewer of many journals. She has also delivered many talks at different forums, and she is also a certified business storyteller. Now, I invite Ms. Arshia Bandari for a talk. Ms. Arshia, please.
Thank you so much. First of all, I would like to thank Sopicon and AIIMS Rishikesh for inviting me for this presentation. I have been I had the opportunity to listen to Dr. Rohit Gupta, and it was very enlightening to understand Delhi from him. And then it was also, I would say, very reassuring to see that the pharmacovigilance, in a way, because Delhi is an adverse event, is being practiced in or is being embraced in clinical practice as well. So my presentation is about pharmacovigilance for patient safety. That how pharmacovigilance is a very important component, not just from the perspective of seeing the adverse effects of drugs, in the pharmaceutical companies, but how it is important to embrace it in clinical practice for patient safety. Before I start, I would like to give a disclaimer on the conflicts of interest. I am a full-time employee of IQVIA, and I have no conflicts of interest to declare. So just to start with, this is very cliche. I would say, everybody says this, first do no harm. But in the long, if you really talk about the healthcare system, the first thing that comes to my mind is our harms, our benefits overvalued over harm by healthcare professionals. And this presentation is about how we need to balance both to be able to bring patient safety into the right perspective through pharmacovigilance. So these are statistics I think you would have heard many times by a lot of speakers, but I would like to talk about these again because it puts into perspective what we are talking about when the burden of adverse events is called out. So if you see among adverse events is among top 10 causes of mortality, it is actually a fourth largest cause of mortality. It causes 2 to 7% hospital admissions in US and Europe. We are taking US and Europe data here because in India, the data is not as refined as it is being collected and established in the countries abroad. It occurs during 10 to 20% of hospitalizations. Expenditure is more than 135 billion US dollars. And ADRs represent a silent but persistent pandemic that is ranked as the 5th most frequent cause of death in developed countries. Indian extrapolated numbers is 4 lakh deaths due to adverse drug reaction and 720,000 adverse events, including medication errors. So the numbers are, quite scary. And that is the reason why it is important to understand how pharmacovigilance can help with patient safety. This is the WHO definition that everybody knows about. Pharmacovigilance science relates to collection, detection, assessment, monitoring, prevention of adverse events with pharmaceutical products. And we'll take each of these, 4 bullets or the points that are in bold in a little detail. And I will talk about how pharmacovigilance is done in pharmaceutical companies, because that's the background that I come from. So to start with, why is, when we talk about pharmacovigilance, one is the preclinical studies that happen and the clinical studies that happen, the clinical trials, versus post-marketing. And this is, a very good analogy that Dr. Jayaprakash from PVPI has shared, and I borrowed it from him. And if you see here, this is the clinical trials, the phase 1, 2, and 3 clinical trials. It's a clear road because it's a very controlled environment where, there are very strict inclusion and exclusion criteria. The special populations may not be included if they are not part of that particular trial. The people with comorbidities are not there. So it's a clear road. The safety is established based on this, but what kind of adverse effects, because there's limited number of patients, maximum 10,000 of patients when we talk about these trials, and because of the limited number of patients, we are not able to see the exact adverse effects that happen. If you, like Dr. Rohit Gupta also shared, the Delhi events are visible in the in after post marketing in the major population, because there's a lot of interaction. There's a lot of factors that are there that because of the interaction of those factors, the adverse effects get manifested. So once the clinical trials are over, the drug is marketed and goes into the real world, which is like this busy street where there are different kind of populations. There is different ethnicities, there are different comorbidities in patients, different concomitant medications that patients are taking together with the medication that is being tested. And that is when, and of course, the number of people that the drug is now getting exposed to has increased manifold. And that is when the real adverse drug reactions can be manifested. But now the question is post-marketing, does post-marketing surveillance happen? Are we, in India, I would say this has increased now after the COVID pandemic. If I talk about my career as a PV professional, I would say when I joined PV, nobody, if I talk to a doctor about I do pharmacovigilance, the first question that comes is, what is pharmacovigilance? How do you do that? And that's in the past. Now there's more awareness. So the point here is that the post-marketing surveillance, the post-marketing side effects can only be established if they are reported and they're observed. So if we see here, this is an overview that I have taken, which talks about what happens when a drug is being developed. This is the post-marketing or I would say the spontaneous reporting and post-marketing reporting that happens in the pharmacovigilance companies, in the pharmaceutical companies, and how these 4 points, that is detection, assessment, understanding and prevention, come into play to establish the side effects or to establish the safety profile of a drug. So if you see here, detection, collection of adverse events from different sources. Now these sources, this is the most important thing. Who are these sources, the post-marketing sources? Because it can be consumers, healthcare professionals, and social media, literature articles, and from the companies, general consumers for that matter. The adverse events can come from anywhere. And I would say this is the prime step which establishes the safety of a drug during post-marketing, because if this detection does not happen, the further process cannot work through. So once the detection happens, what happens in the pharmaceutical company is these adverse event reports are sent to the pharmaceutical company, to the case processing units of the pharmaceutical company, where these reports are processed, and for clinical studies, the narrative writing is done so that these are submitted to the regulatory authorities to assess, continuously assess the safety profile of a drug. Then comes the understanding, where the aggregate data review and signal management is done, so that any new events that are emerging, which were not seen during the clinical trials, can be recognized, and signal management and signal data can be, it is generated so that the safety profile of that drug, that is, can be improved and can be updated. So when we talk about prevention, and when we talk about signal detection, so signal detection leads to signal management. Now, if a signal has been recognized for a particular drug, then it is important that that information is percolated, or that information is communicated very regularly to the people who are actual users of that medication. And that includes, for the first, if you see here, the risk minimization activities are done. There are updates to the summary product characteristics. That's, if you see, there are product labeling inside some of the drug packaging. And because of the new signals that are detected, the updates can be done to the product labelings. Then dear doctor letters, signal alerts that are generated by different regulatory authorities, including in India, PVPI and CDSCO. And eventually, if a drug is seen to outweigh the benefits, the withdrawals are done. So this is the complete life cycle of pharmacovigilance. And if we talk about what comes into the purview, what kind of adverse reactions come into the purview of pharmacovigilance in a pharmaceutical company, it is not just the adverse drug reactions or the major reactions that happen to a patient, which are recognized. It also includes medication errors. There can be, so when we get reports for medication errors, it is not just reports for medication errors that have caused an adverse event, but also medication errors that have not caused an adverse event, to be able to assess or do CAPA on what caused that medication and how such problems with those medications or the drug devices can be mitigated so that these do not happen. Then again, counterfeit medications. This needs to be monitored. Drug abuse, misuse, drug interactions, lack of efficacy, and of course, adverse drug reactions and events that happen. So this comes into, all these events come into the purview of pharmacovigilance. And I speak about it because when we are reporting, it is important that, sometimes we just focus on an adverse drug reaction and not on these allied ones, which should also be reported so that the safety profile of that drug or device can really be refined on. So now I would come to a question here. We all know that pharmacovigilance is important. We all know that it is important for patient safety. But I would like to take you through, this is a report from FDA.gov, and you can find it under this link here. And it talks about that from 1998 to 2000, terfenadine, nibiprazole, estimizole, and cisapride were removed from the US market or restricted in their use as their co-prescription with interacting drugs had resulted in deaths. Each drug had value in the pharmaceutical marketplace and for patients. However, they continued to be prescribed in an unsafe manner, even after warnings from the manufacturer and the FDA. Consequently, the risks associated with their continued widespread availability could not be justified. And this is something we are talking about US, where the pharmacovigilance system is quite advanced and is quite established. There as well, in spite of the constant communication to the healthcare professionals and to warnings from the manufacturer, we could not really save these drugs. If we take the example of a very common example that we all know is thalidomide. So if we take the example of thalidomide, we know that it caused congenital abnormalities. At that time, thalidomide was taken off the market, but because it has quite a lot of benefit, and it is still being used in for leprosy and for other diseases. It was recognized that if thalidomide is not a wrong drug in itself, it was recognized that it is in a certain kind of patients that it should not be used. And and that we know it is contraindicated in pregnancy. So if the contraindications are kept in mind, if drug interactions are kept in mind, the drugs can be safely used. But the issue that we face here is these points, or during the prescription, sometimes this is missed, and that keeps building on the adverse event burden. So I talked about thalidomide here. I would like to talk about valproate, and this is a report from AIIMS, where PRAC, that is the risk assessment committee, a pharmacovigilance risk assessment committee from AIIMS, it recommended new measures to avoid valproate exposure in pregnancy. And you would be surprised to know that the scale of the issue and harm caused by valproate is bigger than thalidomide. So thalidomide is probably not being prescribed during pregnancy, and this is a report from EU that despite warnings and communications to healthcare professionals and patients, 1 in 5 women taking valproate in EU are unaware of the risks of taking this medication during pregnancy. And there were a lot of different new measures that were rolled out by AIIMS, and the assessment is still going on if it has helped. So if we really talk about, so this is from EU. When I read this report, I did a little survey I went to chemist and I asked for valproate prescription. I did not have the prescription, but I just asked for it. I was given the drug. I did not buy it, but I was given the drug. I was given 3 different versions, as in from 3 different companies. One of the companies had the black box warning on pregnancy. The other 2 companies did not have that warning. And and that's where we are somewhere lacking in communication or lacking in product labeling, where it is very, very important, because this it's very important that it's also a Schedule H in under prescription drug, but still it gets prescribed, or it gets available in our pharmacies without a prescription. There still needs to be done a lot on the correct product labeling and correct monitoring of valproate in India when we talk about being prescribed this medication during pregnancy. So these are a few examples I wanted to take where I am trying to emphasize on that where we are still lacking, and where, and these are only 2 examples that I took, but there must be a lot of other medications that are being used unsafely, and that needs from our regulatory authority and from the healthcare community, that needs a lot of surveillance so that patients are not harmed. So if we see here, what is the purpose? Why pharmacovigilance? It is risk mitigation, prevention of adverse drug reactions, timely identification and management of adverse drug reactions, and patient safety. So the first step to all this is detection of adverse drug reactions. And if we talk about who is the most effective ears and eyes of a successful pharmacovigilance system, I would say it will be, and that's a question that I would like to pose to everyone here. What do you think are the most effective eyes and ears of a pharmacovigilance system? I would be grateful if somebody could off-mute themselves and maybe answer this question.
They are healthcare workers.
I agree very much. It is the healthcare professionals, because healthcare professionals have complete understanding of medicine and the disease processes, and to be able to understand and to find whether an adverse drug reaction has a relation to a drug or not, whether it is drug induced or is it a comorbidity. That kind of understanding. So when we talk about gold standard reports in pharmaceutical companies, what come to us adverse event reports, we've always been looking for the confirmed reports, medically confirmed reports. Though information from patients, we cannot undermine the importance of information from consumers as well, because they are experiencing the adverse effects, and they know their body the best. Therefore, though the most effective eyes and ears of a pharmacovigilance system are HCPs, it is important that reports do come from everywhere. And when we talk about HCPs, it is important not just to recognize these adverse effects and probably treat them on their own. For a successful pharmacovigilance and to build the safety profile of a drug, it is important that these are reported to the regulatory authorities or to the pharmaceutical companies, so that the signals, if we are seeing some, are getting recognized. If that is not done, then it is always, I would say, everybody is working in silos. It has to work in unison if we really want pharmacovigilance to work for patient safety. Okay. So this is a very interesting journal that I studied, where it talks about what future healthcare professionals need to know about pharmacovigilance, and it talks about the house of PV in clinical practice. And if you see here, this is this diagram shows the house of PV. It talks about the understanding the importance of PV in pharmacotherapy, and that makes the base of this house. It is if we understand and we recognize the importance of PV, we will be able to manage adverse, these are the motives that makes the walls of this house. First of all, to recognize the ADRs, to manage the ADRs and to prevent the ADRs. And if we see what is the roof of this house, it is reporting of these ADRs, which is very, very important, because unless the reporting is done, it will always be in silos. And so if you see, when we report adverse drug reactions, it is a mandate that comes from the regulatory authorities is that you do not need to ascertain the causality to be able to report an adverse drug reaction. It is not important that if it is related, that is only when it should be reported to the regulatory authority or to the pharmaceutical company. Because there is an extensive process of signal management, signal detection that happens, causality assessment that happens, that is important for that creates the causality or does the signal detection for that. So therefore, it is important that the ADRs are reported frequently. Now, I'll come to this one point here, that diagnosing through, am I audible?
You're audible, Ms. Arshi. You just have 2 minutes left. Can you please wind up in 2 minutes, please?
Sure, I'll do that. Thank you so much.
Thank you.
Yeah. So next, what I wanted to talk about is the prescription cascades, and this is a concept that I would say everybody here, because the healthcare professionals are here, and you would understand better than me. But I think this is what is the need of the hour, that the prescription cascades are recognized in time, so that the adverse effects can be recognized. For example, if we take the example of this patient who has depression, she was given medication 1, bupropion, for depression, which had a side effect of insomnia. Medication 2 was given for insomnia. It was not assessed whether it could have been caused by a prior medication. And then, new side effects. And it's always a cycle that happens. And it is important that, this is recognized as soon as possible, so that the patient is not given medications which are not required, which in any case are going to cause more side effects. I'll go forward. I'll go forward on some of my slides. This is something, factors that affect ADR. And this is very beautiful. It was taken by Dr. Rohit Gupta in Delhi, that how different factors are relevant when that which patient is going to experience an ADR. So I'll come to this slide, and this is one of my last slides. Systems approach. So if we see this is an article that I wrote for Uppsala Reports, that is the WHO publication. And I talked about how it is important now to diagnose and prescribe through the pharmacovigilance lens. As we talked earlier, it's a universal phenomenon that in the healthcare community, sometimes we value the benefits more than the side effects or the adverse events. And it is important that there's a change or shift in this thought system, so that the adverse event burden can reduce. So my question here is misdiagnosis of an adverse drug reaction as a new ailment by clinicians, just a human error, but a lapse of the system. With the magnitude of data on adverse event reactions and drug interactions, is it possible for clinicians to keep track of all AEs and interactions and be conversant with latest literature on a regular basis? And if we take systems approach there, then it's very difficult. So what can be the solutions towards it? So there's a strong need to support clinicians with tools and pharmacovigilance. From the technology perspective, drug interaction trackers like Hippocrates and medical letter can be used as powerful aids. Quick searches in databases like VigiAccess and UDRA-Vigilance can also inform healthcare professionals of emerging signals. Then use of clinical decision support systems, regular consults, medication and adverse event review by pharmacologists during hospital rounds, and hands-on experience with pharmacovigilance data in ADR monitoring centers during internships and medical training could also assist future clinicians to appreciate the significance of pharmacovigilance in clinical assessment. With this, I would like to wind up my presentation. Thank you so much for your patience and for listening. If there are any questions, I'm happy to answer. Thank you.
Thank you, Ms. Bhandari, for this enlightening talk. So now the house is open for the question answers. Are there any questions for the ma'am? So Ms. Bhandari, that was a very excellent talk. And you very well explained the concept of prescription cascade, which we see day in and day out, especially in the elderly patients. And it was really enlightening. And rightly you said that clinicians cannot cope up with the fast changing things which are happening in the pharmacology industry. So it's a high time that the pharmacologists and the clinicians join hands for the patient safety. That will be the good thing for all of us, and that is the solution of the present era. Thank you for the talk.
Thank you so much.
Thank you, ma'am. Due to unavoidable circumstances, Professor Ashok Dubey will remain unable to present his talk today. So we move to the felicitation. On behalf of organizing committee, I express my heartfelt gratitude to Ms. Arshia. Ma'am, we'll be sending your memento via post.
Thank you.
May I call upon Professor Shailendra Handu to kindly felicitate chairpersons of this session, Professor K.H. Rita and Dr. Meenakshi Dhar. Thank you, sir. Now we'll take a short tea break for 15 minutes and then we'll gather back. You may kindly proceed for tea. Welcome back, delegates. Now we proceed towards fifth and last session of day one of Associate Professor, Internal Medicine, AIIMS Rishikesh. With the next talk, we hope to enhance your understanding of pre-marketing studies in drug safety assessment and the critical role in ensuring patient safety. I request our courteous chairpersons to preside over this session. I welcome you, sir.
Evening delegates. Starting the session on artificial. So this session will deal with artificial intelligence in drug use, and I'd like to introduce speakers.
Good afternoon to all. The first speaker for the session is Dr. Soibal Mukherjee, the consultant, and I welcome sir to the dais for the first presentation. And he's heading medical affairs and the clinical research at Pfizer Ranbaxy and in the Quintals Asia. And he was also founding president of ISCR and member of many committees like OPPI and FICCI. And also he's a member of advisory expert committee in the BIRAC ICMR. And he has received many achievements, awards, including Clinical Research Lifetime Achievement Award for the exemplary Dr. Soibal.
ये full screen ही है full screen इस तरह से नहीं है पता नहीं क्यों नहीं आ रहा क्यों नहीं आ रहा ऐसे आ रहा है हाँ यहाँ पे तो है पर ये वो नहीं कर रहा शो नहीं कर रहा है ये सिर्फ read only है क्या
Before sir starts the program, I want to tell something about the artificial intelligence utilization in this drug safety or more often in the pharmacology aspect, because I am also nodal coordinator in AIIMS Rishikesh as part of AI Center of Excellence. Ministry, Health Ministry has healthcare sector, how AI can be helped in our daily practices and also daily monitoring and also helping the patient as a education aspect also, because in major problem in Indian setup, you all know, because as a doctor, I feel when I am seeing patient, the illiteracy about the health system among the patient population. and how that can be tackled, because without that, definitely the major development will be less. For example, the drug safety you just see. Many patients are having adverse reaction that no one is reporting. Majority. Like you can take the public aspect, and also you can take the in the hospital stuff also. So how AI can help, that we listen in this sessions. Welcome, sir.
Yeah, so thank you for the introduction, chairpersons. Unfortunately, we are having a little problem with the slides. They're not going on to the full screen mode. So we will continue with this. I think there. And the title of my presentation is Drug Safety Assessment Pre-Marketing Studies. So there's not much of artificial intelligence in drug safety assessments and pre-marketing studies at the present time. We don't know what will happen in the future. We might have some artificial intelligence come into this in future years, but I find it somewhat unlikely because, you know, for artificial intelligence, you require data, large amounts of data of the same kind. When you're discovering and developing medicines, drugs, each drug is a new one. And therefore, what applies for an earlier drug that was developed, say, 2 years ago or even yesterday, does not apply to the new drug that might then be developed. This is just my thought. Maybe in future, we will have some applications at least of artificial intelligence that will come into pre-marketing studies, certainly. Nevertheless, I want to talk to you. We have just 20 minutes, so I've kept it a very short presentation. I have in all 9. For discussion. So let me get to my first slide. So the background of this presentation is that this applies to all new molecular entities that are being developed. So, you know, usually we are talking of medicines, drugs, and pharmacovigilance once the product is in the market. However, my presentation is about before the product comes to the market. So it applies to all new molecular entities, new indications, new routes of administration, and new formulations. Now, the first one is self-evident, all new molecular entities, but we tend to forget that a majority of new or phase 1 studies or developmental studies that often happen with tweaks, new formulations, new indications, new routes of administration. There are new kinds of formulations that are being developed which deliver the drug better, and you have to test those as well. This applies to preclinical studies, which provide the basis of the clinical development program. Early studies that are done in this continuum from phase 1 through phase 2 to phase 3, the early studies must be designed for sensitivity for unexpected toxicity. What that means is that you do not know what you're going to see. Of course, there have been preclinical studies already done, and there is some idea of what kind of toxicity is happening in animals. But animals are not always a good predictor of what will happen in humans. So what might happen in a phase 1 rising dose study, multiple dose or single dose, is sometimes going to be surprising. And these studies must be sensitive to this. That means that you have to do baseline for almost a wide variety of indicators, including biomarkers, liver injury, hepatic injury, renal injury, and if there is any indication of any other kind of injury that might happen, you have to screen your volunteers for it first, and then continuously through the period of the early studies, measure those for widespread use. So the initial studies don't do that. It's the later studies, the phase 3 studies, and phase 2 is somewhere in between. They are for the assessment of benefit risk ratio. So the drug might be beneficial, but if it has a risk profile which is not acceptable, then obviously the drug won't move forward commercially. So, this is a very tricky thing there. So what kind of risk is not acceptable? Obviously, if there are big risks to life or limb, then obviously that is not acceptable. But in a commercial development process, you have to also see what is already available. If you have a medicine that is being developed, which has same or even better efficacy than existing products, then obviously from an efficacy point of view, that's acceptable. But even if it has a slightly lower safety profile, that is worse safety profile, it might become unacceptable because your market becomes smaller. You might convince some physicians and some patients to risk ratio. Comparison to existing therapies may be both relevant, as I said, and risky from a commercial viewpoint. So while I say that you have to look at what is already there in the market and in that way assess the benefit risk ratio and see whether that is comparable or better than what is already there. However, it's a risky proposition. Often when you look at new studies for NCs that are being done, you'll see that the comparison is with placebo. The comparison is not usually with active drugs that are already being used for that indication. And that is because if you do that comparison, and if that comparison even is slightly worse for your product, then the development team finds it very difficult to convince their managements, and the managements also then, there's a lot of commercial stake in this entire thing, because then the share prices of the companies go up and down depending on how your product is doing in your clinical trials. The results have to be transcribed so this is the last part and very purely process point of view, the purpose of making sure or of documenting safety and toleration in phase 1, 2, and 3 studies is to understand what should go into the product document. So there is the investigator's brochure. So after you do the phase 1 studies, whatever safety pharmacovigilance information is garnered has to be transcribed into the investigator's brochure that will be given to investigators of the phase 2 studies. And from that to the phase 3 studies. So it has to go into the IB. The results of this have to also get into the product label that you will submit to the regulator at the time of the submission of the NDA. And the regulator will look at that and say, no, we've reviewed your studies and you have to put these words in there and this line has to be there and you cannot have it for all groups of patients. This group of patient has to be excluded from the indication because these patients seem to be at greater risk of side effects and things like that. And finally, after every study, you might need to modify your informed consent document as well, because you must tell patients. So with that background, when you get into the drug development process, the preclinical studies, there are a few questions that need to be answered. So the studies that you will do need to answer these questions. What are those questions? These are, what is the safe range of doses that can be tested in humans? This is a question that is asked in the very beginning. What is the safe range? Because when you're going to give it to either volunteers or patients in phase 1, phase 2, and phase 3 studies, you need to know, particularly in the early studies. In the later studies, of course, you have kind of come down to 1 or 2 doses, or maybe 3 doses. But in the early studies, you have no idea where your dose is going to end. So you need to have an entire range in front of you and see up to what level you can go. And also when you start in the phase 1 studies, you have to provide a good safety margin so that some unexpected side effects don't happen at a high dose. The second question is, how do the preclinically identified risks translate to humans? So if you've seen side effects in rabbits or do they not happen, and are they not relevant for humans? All answers are possible. What is the relationship of the dose to the pharmacokinetics of these risks, to these risks? So is this, are these risks related to the pharmacokinetics? As the dose goes up and the plasma concentrations go up, do these risks increase, or are many of these risks or some of these risks idiosyncratic? Even a small dose can cause that problem, as happens with allergic reactions and stuff. Which doses provide the best benefit risk ratio? And this you will see in usually phase 2 studies, where we do dose finding. And then what factors influence the risk profile? Does the risk profile get worse with aging patients? Do they get worse with patients who are compromised in some other way? Do they get worse when you give it together with a usually concomitant use medication? So all those questions need to be answered, and these clinical trials will answer those questions, and you must be focused on some of these questions as you design those trials. So phase 1, the use of a safety factor. Low dose, of course, we know the calculation for safety factor, and we apply that safety factor and give it to maybe just one volunteer to begin with, and then increase it. That one dose goes on to, you increase that dose and give it to one more patient, and the lower dose then is then given to 2 volunteers, and so on. Determination of the maximum tolerated dose and associated pharmacokinetics is another thing that is studied during the phase 1 studies. So you increase the dose, and as you go on increasing the dose, you finally see at what dose side effects tend to begin to happen, and what is the maximum level up to which you might be able to give the drug. So that gives you the range in which you can play during the later studies. There's the correlation of safety and toleration with pharmacokinetics and biomarkers. This is very important because as you look at the toleration profile and the safety, and if it is related to the pharmacokinetics, then you can easily predict what will happen in the next cohort as you increase the dose. Also, the pharmacokinetics itself may be linear, or it may not. Basic safety and first draft of warnings and precautions and contraindications are written at this stage. So after you finish a phase 1, which is the single ascending dose, the multiple ascending dose, and some special studies to look at drug interactions or food effects and things like that. When you are done with these studies, you are in a position then to begin to write your product label. In that label, you will be writing the basic safety paradigm for that product. So you would say that these are the main warnings, precautions, these are the contraindications, and those things should be clear to you after doing these phase 1 studies. So the phase 1 studies have to be designed to answer that question. Evaluation of overall toleration profile. It gives you, so that is also another thing that you put into the product document, and evaluation of the drug interactions that may be relevant. So when you are, so these are done, the drug interaction studies are done usually that are expected to be given concomitantly. So if you have an anti-diabetic product, and we know that there are so many anti-diabetic products in the market, some of them will be given concomitantly with this one probably. So you must already assess that in special studies, interaction studies, which are still phase 1, even though they sometimes happen concurrently with phase 2 or phase 3. Then as you come to phase 2, this is the stage of proof of concept. That is whether the disease, because in phase 1, usually, generally speaking, phase 1 is done in volunteers, there's no disease there. So when you come to phase 2, you're taking patients who are ill and you're trying to cure the disease. So whether it actually cures the disease in humans is a very, very important question. About 50% of drugs, more than 50% of drugs fail at this stage, because even though animal models tended to indicate that the drug will work in this particular disease, when it comes to humans, you find that it doesn't, or it does at a the dose. So in the phase 1, you've got a whole range up to what level it will be tolerated, and you also have the pharmacokinetics. Now you marry the pharmacokinetics to what is the drug concentration that you've seen in vitro studies that is required to engage with the target. And you see at what pharmacokinetic level you will that is most likely to happen. And accordingly, you choose based on an evaluation of the benefit on the one hand, that is the pharmacokinetic levels and what can give you the efficacy, versus what side effects you have seen in phase 1 studies. And you choose maybe 2 doses or 3 doses for a phase 2 study. So the safety that you have assessed in phase 1 therefore becomes very important for you to choose the doses in phase 2. And then in phase 2, you go ahead, correlation of risk with the degrees of morbidity. So is this drug one in which if the patient is more severely sick, then is the interaction potential with common concomitant medication, as I said, second draft. So at the end of the phase 2 studies, you can have then begin to have the second draft of the warnings, precautions, and contraindications. Now, after phase 2, you have a very good idea, because now you have given it to patients. Patients will respond slightly differently from a safety and toleration point of view than healthy volunteers, because the healthy volunteers don't have the disease, and in phase 2, you have sick patients there. So now you can fine tune the warnings, precautions, contraindications, the wordings, the phraseology that you use will become different. And then at this stage, you also write the first draft of the toleration data. So safety and toleration, what is the difference? The difference is when we talk of safety, we are talking of major side effects, usually serious adverse events. On the other hand, toleration means some small things which are tolerable, but the patient is uncomfortable, and therefore, you will often see a whole list of insomnia, label. They must be mentioned, because you never know to what extent the drug was contributing to it. So the first draft of the toleration data then goes into the product level label at this stage, and the evaluation of drug interactions as may be relevant. So these patients are sick patients, so they are getting actually usually the concomitant medication that will actually be used when this drug goes into commercial use. So those are some of those things that from a pharmacovigilance point of view that will happen in phase 2. And then we come to phase 3. And the phase 3 really, by the end of phase 2, you get a very fair idea of what this drug is all about. What are the safety concerns here, how it will be tolerated by patients, and whether it works or not, and how well it works. So what is phase 3 for? Do you require a phase 3? Of course, you do require a phase 3. We know that phase 3s are the biggest studies that are done. They are often in thousands of patients. There are certain numbers that studies and the pharmacovigilance information that you get in the phase 3, that you finalize the product label. Some of these are contentious issues. So you put in something in the product label, the regulator comes back and says, no, your studies don't show that, or your studies show something else. So you must add these lines, you must put this extra warning, this precaution, this one is a contraindication, you can't have this drug given to these patients. All of that gets finalized at the end of phase 3. So full safety and toleration data, including rarest adverse events, must come into the label after your phase 3. Final drafts of warnings, precautions, and contraindications for the new drug application. Directions for use in patients with compromised organ function. So now in phase 3, you would have patients probably with some degree of renal compromise or hepatic compromise or some other type of issues. And you can have directions for patients for these conditions. Detailed section on drug interactions and things like alcohol use, use of machinery, directions for driving, all of these are required to be there in your product label. Data from the patients that you include in phase 3 studies. Advise on the handling of acute side effects, if there are any with this drug, and the precise place in therapy. So whether it will be used as a first line, whether you are recommending it as a first line, or whether and a lot of that depends on pharmacovigilance. It depends on, the drug works. You found that in phase 2 it works, and that's why you took it to phase 3. However, you need to define where in therapy it needs to be placed. And then the phase 3 and the entire application also determines the mandatory post-marketing data that the regulator will ask you to generate. So while on the one hand, you have done these studies and pre-marketing is pre-marketing, but pre-marketing also determines what you will be asked to do post-marketing. So the risk management plan for a known serious adverse effects, special studies to investigate certain adverse effects that are in focus, open label studies or extensions to generate additional So I think that is almost my last slide. This is my last slide. So now this is basically what you do in pre-marketing studies, and that determines what comes into the label and how this drug is going to be used in patients finally. What you can promote to doctors when you're promoting the drug depends on what you've done on these studies and what has come into the label. In most countries in India, we don't have very strong regulations about these, but in the developed world, there are very strong regulations on what you can promote and what you cannot. And whatever is not in the product label and has not been referenced in good quality clinical trials cannot be promoted. You cannot have them on your brochures and your marketing material. And the brochures and marketing materials in the US have to be approved by the regulator before you use them. And they will look at this product label, which is a distillation of all the pre-marketing studies that you have done that comes into this product label, based on which you'll be able to market your product. So important because if you don't follow the right scientific principles, and I say this because unfortunately in our country, drugs are approved without proper studies. Even today, there are formulations that are or drugs that will be approved by our regulators here in an open, uncontrolled 100-patient study. Sometimes these are drugs which we don't bother much about, drugs or nutraceuticals or devices, for example. We don't bother that much about them. But what tells us that there might not be risk associated with some of them? So the need for a control group, the elimination of bias through blinding and through randomization, the establishment of a strong baseline, which is relevant so that you can see what changes happen during the trial, control of other variables, dropouts, and how you handle loss to follow-up and unavailable data, adequate size of the safety population. So while the efficacy population might be as small as 400 or 500 or 600 patients, the safety population has to be 50. Vaccine products, but other regulators don't allow that. And there's a minimum number of the safety population that you are required to achieve under the ICH guidelines. So that brings me to the close of my presentation. Sorry if I have overshot my time by a few minutes. If we can have some time for questions, then I'll be happy to take them.
Thank you, Dr. Shoival Mukherjee, for your wonderful presentation on pre-marketing safety assessment. I now open the floor for questions, if any. I have a question, sir. I mean, you know that underreporting is one of the main problems for adverse drug reactions. So, like while you are picking up adverse drug reactions in pre-marketing, safety assessment.
Could you speak a little louder?
Yeah. So, is there any method of, method like predictive analytical methods to pick up?
One is the pre-marketing studies and then the post-marketing. So whether there are some analytical methods or we can probably do it with some artificial intelligence, a lot of things are happening out there. They are developing new tools to look at how a drug is interacting with different molecules within the human body, the milieu in which the drug is working, and trying to, from that, make modeling algorithms which will predict what kind of side effects you are expected to see. But there's no telling whether they will actually happen or not. So as far as I can see, for the predictable future, we still need to have clinical trials before we put a product into the market. Unless we do that, these models, they might predict certain things, but you can never, unless you can't take the risk of putting a product into the market based on a model. Maybe we will come a few decades later to a stage where artificial intelligence of modeling studies, but I don't see that in the near future, at least. Innovative molecules. So let me come from the international to the national. Yeah, so I'll make an estimate of the national scene. The international scene is about 20 to 30. Sometimes it goes above that. The national scene will be about half or about a third of that. Now, of course, we have product patents these days. Earlier, we didn't have that. So when we didn't have that, everybody would put it they saw the market, particularly in the 90s. Before that, we did not have the capability. So there were not too many companies that would begin to do this. But in the 80s and 90s, there were enough number of generic companies, and we still did not have any product patents. So everybody there would be, kind of 20 companies that would be researching the same molecule and wanting to put it into the market. And every product that had a market in India would be introduced. Right. So after the 90s, what happened is the Indian clinical trials market also opened up. So global trials started being done in India. So Indian centers, maybe 5 centers, 10 centers in India, 10 sites would be part of maybe 200 sites worldwide. And India would contribute to that data. And the Indian regulator has said that if you do a sub-analysis of just the Indian data and show it to us, and if that number is more than it's a respectable number of patients, then we will look at that and we will waive the study that you have to do in Indian patients, because Indian patients have already been exposed. Yes, sir. Submitted to the regulator at the IND stage. So the quality data of the product is already IND, meaning before you can start any clinical trial. So even before you can start any clinical trial, you have to submit the entire data of what is there in the product, including the complete total full impurity profile. So if there are nitrosamine impurities there, then they have to be reported. If they are at a level which is higher than acceptable, then the regulator will take a call, and it depends on how much of it is there, that they will either disallow it and say you have to reduce it, you have to purify this product more before you can use it in humans, or if it is within the acceptable range, small amount, sometimes trace amounts are there, which are well within acceptable range, then they might allow it.
Okay, then this, when designing the dosage for a drug, is it that impurity content also they'll be designing during the clinical trial? Like for example, nitrilo triacetic acid impurity. Somewhere if you are using disodium syrup, recent incident took place when Gambia, around 300 children so far death occurred.
Yeah, so so clinical trials don't have anything to do with this particular issue.
No,
so even after
sir, they are saying ki due to diethylene glycol.
Ethylene glycol. Yeah.
Diethylene glycol present more than the permissible limit in the cough syrup has caused this death, but so far nobody has identified that. I mean, root cause investigation, even this drug regulators or in India.
Sir, I'm not able to
Yes.
hear the word very clearly, because I think there's a little, but let me tell you that this thing about diethylene glycol contamination in cough syrups, it's such an old issue. Such an old issue. It has it goes back to the 1930s. In fact,
No, sir. It just, I think November, Gambia, the cough syrup supplied by the Indian manufacturer caused around 300 children's death so far.
batch of cough syrup that came from China. I mean, the diethylene glycol contamination came from China. And then we have had that same issue so many times in India. We have had it in Bangladesh, we have had it in Pakistan. And still we can't solve this. It has nothing to do with clinical trials. This is the contaminant.
Yeah.
Yeah, and it is being, the contamination is coming from industrial solvent that is there. And that contamination is, of course, there's no connection to clinical trials because diethylene glycol should not be used. It's triethylene glycol. If I don't remember exactly which ones it was, but I think diethylene glycol gets confused with triethylene. Contamination happens. One of them is toxic, particularly to children, renotoxicity. So that is the issue. And clinical trials have no solution to that because obviously and you will use uncontaminated pure product in your clinical trials.
Okay. Thank you.
Okay.
Remarketing safety assessment. Thank you so much. Our next speaker for this session is Dr. Anand Srinivasan. Dr. Anand Srinivasan is additional professor in pharmacology at AIIMS Bhubaneswar, and he has 54 publications. So I hand the session to Dr. Anand, please.
I thank the organizers for providing me this opportunity to talk about one of my favorite subject. The credentials for me to talk about this subject is because I'm pursuing a PhD in artificial intelligence in health sciences as a joint program between IISc and AIIMS Bhubaneswar. So with that, I'll move to my presentation. So I'll try to keep things very sweet and simple because it's just a 15 to 20 minutes presentation, and I'll be just giving an introductory overview of what artificial intelligence is and how it's useful in healthcare. However, when it comes to artificial intelligence in pharmacovigilance and drug safety, we already have one more presentation after that, either today or tomorrow. Okay. So when we think about artificial intelligence, many have this idea in their mind, some magic is being happening, in the back. What's really happening in the background is this simple mathematics. Okay, so this is what this depiction is the core of artificial intelligence. So this is the neuron of artificial neural networks. Okay, so we have cell body where all those functions occur. We have the inputs in the form of X's, that is matrices and weights, and we have output. Okay, so this is hardcore. Even the complicated chat GPT uses this one in its core in the background. So what's core of artificial intelligence? Of course, without data, your artificial intelligence does not exist. Okay. Now, for anyone who wants to seriously learn and practice artificial intelligence, there are two pillars which we need to be equipped with. Okay, to be a serious artificial intelligence researcher, not in a superficial format. So one is want to learn and implement artificial intelligence is the first one is we need to be comfortable with the calculus, because whenever we perform various operations like linear descent during optimization or back propagation in artificial neural networks, calculus really is essential. The second is the probability and statistics part from the mathematics, because almost all of the artificial intelligence algorithms from which the models are created can be viewed as a form of a Bayes theorem. Okay. And the third one is, so we need to be comfortable with the linear or the matrix algebra. So that is how the data is handled within, say, artificial neural networks or any other algorithms. Then the next thing is the programming tools which we need to be familiar with for performing artificial intelligence research. The first and foremost is the famous tool which is used these days is Python. And the second one is even R can be used for generating AI models. And there are other tools like we have MATLAB. And if you want the model to perform very well, then we need to go for languages like C++ and Scala and all. Okay. Now, whenever there is a data analytics part in a conventional way, what happens is when we look for a correlation between a parameter change and the effect, we do perform use simple statistical tests like t-tests or something. Okay. So this in the conventional approach, this parameter change is looked upon in a separate individual fashion. Okay, is in an isolated fashion, I'll call it. However, when we talk about AI approach, so this parameter change is looked upon as an interaction between the other factors, which in healthcare. Okay, by extension, pharmacovigilance also, because this is how the biological interaction look in a human being. Okay, our human being is actually, humans are actually a set of biological molecules interacting with one another. Each and every molecule is actually interacting with one another and comes to an equilibrium. Okay, so a perturbation in one molecule can either dissipate or can lead to or spiral into a larger perturbation in some seemingly unrelated end. Okay, so to understand this concept of biological interaction, the conventional approach will never work. We need to look into the artificial intelligence approach, because even the ADRs from giving the drugs, it doesn't depend only upon your drug. It also depends upon the various patient factors and even certain other factors like social factors also, if we look it into a broader perspective. Okay, so that is why AI is useful in healthcare research. So now what happens in AI research? What happens in AI research? Okay, so when we start doing some artificial intelligence research, or when we want to practice artificial intelligence, the first and foremost thing to be done is model development. Okay, without developing a model, we cannot predict something. Okay, so we'll talk something about this model development. So we use this approach called machine learning, okay, for developing models in AI research. Okay, so when we talk about the machine learning part, we can approach this machine learning as first is unsupervised learning, and the second is supervised learning. And our healthcare research usually belongs to the supervised learning part. So basically, because when we have the data, we know that whether the ADR has occurred or not. So that becomes a supervised learning, because we know the actual labels or predictors. So sorry, not predictors. We know the actual labels or the effect. Okay. However, in unsupervised learning, we don't have a scenario, or we have a very little number of scenario where these labels are present. Okay. So this is what how to do about it, go about it. So we have labeled data. Okay. Then this one, many of you might be aware. So we split the data into testing data and training data. Okay. And we use the testing data for model development, including the usage of validation data set for hyperparameter tuning. And then we use the test data to check for the model accuracy. Okay, right. Now, so when we are talking about the data, what are the sources of data in healthcare research? So we can broadly categorize the sources of data in healthcare research into two major parts. So one being the image based, the second being non-image based. The artificial intelligence in image based has actually a lot of work compared to non-image based artificial intelligence, because this image based health application of artificial intelligence in healthcare is just an offshoot from the image based presence or absence of disease from even an X-ray or CT, or even the generative models can even locate the position of the lesion in MRI or CT, something like that. Okay. However, non-image based data will be the one mostly used for artificial intelligence, especially when it comes to pharmacovigilance. So what are the sources of this non-image based data? So one will be clinical data, then the lab based data. When we talk about the clinical data, it is the general patient clinical data such as demography, history, clinical findings, other things like drugs used, drugs prescribed, all those things. Then we may have signal data from ECGs, EEGs, all those things. Okay. So when we talk about the lab data, which can be used for modeling in AI, so one can be omics, be it genomics or metabolomics or proteomics. Yeah, proteomics and metabolomics are just a broader view of viewing our we have the data of pathological data in the form of slides or gradations or whatever it is. Okay. Now, when we further look into the domains of healthcare in AI research, if we look into the various publications or even the applications, very few applications which are developed, okay, it broadly falls under two categories, that is diagnostics and therapeutics. Okay. Basically, both these approach actually focus on precision medicine. So if when it comes to pharmacovigilance, if we want to, predict the presence or absence of ADR, it can fall under this diagnostic part. However, if you want to use the data for pharmacovigilance for dose modification, it comes under therapeutics data. Basically, both serve a common goal of precision medicine. Okay, so we have the data and all, and we are in the process of model development. So usually what are the various algorithms used for AI model development? Okay, so first is the then we have various other algorithms like decision tree, K nearest neighborhood. We have X gradient boost. Then we have support vector machine algorithms. Then we have artificial neural network. But these days, practically AI is synonymously like practical associated with artificial neural networks. After the after the practical utilization of artificial neural networks became easier, relatively easier, AI has been completely taken over by artificial neural networks compared to other algorithms. Okay, I'll discuss certain examples here where AI has been used for healthcare research. So we have this publication where it talks about detection of intrauterine growth restriction using metabolomics data. Okay, so basically they had samples of metabolomic analysis of cord blood serum. They had samples from 40 cases and controls. They used the support vector machine algorithms. It's a good accuracy. Okay, so this is one of the publication where AI has been used for predicting the intrauterine growth retardation. And there is another publication here where metabolomics data was used to diagnose malignant mesothelioma. Okay, just from metabolomics data. This was published in Diagnostics. So there were samples from 25 cases and 32 controls, and they had used random forest algorithm. So here, based on the various predictors, they have found that the accuracy is around 80 to 85% for certain models for diagnosing mesothelioma. This is without the imaging data and all. Okay, imaging or your pathological samples, something biopsy samples. It's all from the blood sample data only. Then this is one thing which is relevant to pharmacology, wherein there is a model called, there's this very recently developed model called that is ordinary differential equations over neural networks for analyzing the pharmacokinetic data. So if we find here, we can observe that the prediction of concentrations of this trastuzumab in patients with neural ODE was much better than the other algorithms like nonlinear mixed effect modeling or long short-term memory networks or light gradient boost machine algorithms. So, okay, so what about the AI and pharmacovigilance? Okay, so the concept of the algorithm which is actually used in pharmacovigilance is called natural language processing, which is actually the same algorithm which is used in your ChatGPT. Okay, so what happens is natural language processing is nothing but an architecture using artificial neural networks, which is which has been trained on electronic health records of the hospitals to detect scan the text data and look into the various clinical features mentioned, and it will also look into the drugs prescribed and see whether some sort of relation is available. Okay, so later on, whenever a newer data is presented, then that NLP algorithm, the model generated from this NLP algorithm will provide a signal. Yeah, this drug is causing some ADRs. And this has been deployed in certain hospital and many hospitals in the Western countries, where they have used this natural language processing models developed from this natural language processing for detecting AI. Might be the next session can discuss about this in a little detail. So, all this artificial neural networks or support vector machines and this concept of artificial intelligence is not at all a new concept. It's there for many decades, or even you can call it as half century. And the concept of artificial neural networks dates even back further. Theoretically, it was always available, but why now? Why so much of fuss about artificial? With the computational power available, just to train the data, it would have even taken years. But right now, even my laptop can do it in, if not in minutes, it can do it in hours. Okay, then because right now the computer power and technology has grown exponentially, AI has been used for practical applications. Okay, and not only that, as I told previously, we need data to build AI models, and the data for pharmacovigilance or for ADR prediction is electronic health data. Okay, so without the availability of this electronic health data, the concept of electronic health data is a relatively recent thing. So that's why in the recent days, we are hearing a lot about the use of artificial intelligence in almost every area of health sector, including the pharmacovigilance. However, of course, there are certain limitations because, as Dr. Shwebal already mentioned, without a large amount of data, your model and research interest. Then we have the logistics and expertise issue and the cost issue. That's all about the use of artificial intelligence in health sciences. Thank you.
Thanks, Dr. Anand. If there are any questions from the floor.
Sir.
So Anand, you just mentioned that large amount of data is required. Now, when we say large, how large do we mean? Is the data from one hospital sufficient, or do we require data from many hospitals, or do we require data from the entire country? What is the scale?
So the scale, actually, there is no clear-cut definition, sir. It depends upon the application, and also it depends upon the predictors or the variables which we take to build the model. If the variables which we take to build the model has a good scientific here is, it is at least in thousands. So that can be considered as some cutoff point, but it's not true. However, if we are just just searching in the dark without any proper scientific rationale, then we need lakhs and crores of data. So basically, so that's why that's where the later on, definitely this AI is going to it will be complementing the doctors, and might be in certain other fields of health sector, it can replace the jobs. But the thing is that for successfully implementing the AI models in practice, people need to be trained to look into these data properly so that they can develop the AI model successfully. So that sort of training has to be provided these days to skill up, basically. So my point is, it all depends upon the data and the human understanding of that data, like how this data can explain the prediction. Yes.
In one hospital, it's easy to manage that, but
Yes.
maybe a hospital chain also can do it. A
Yes.
private hospital chain or
Yes.
whatever, or all the AIIMSs of the country can pool together the data.
Yes.
But there is a limitation of how much data we can have in this country.
So this one, sir, this natural language processing is immune to that.
Okay.
Is immune to that because it just scans the words. So what your research actually looks into various predictors and see whether the response is there or not, something like that. So where when natural language processing is not used, then whatever you have mentioned actually comes.
Yeah.
Actually, one interesting thing I have to mention here is, see, like theoretically, artificial intelligence model can be developed in small data also. But what happens is there is this concept called overfitting. The forthcoming newer data, then the accuracy drops down. So that's the issue with the AI.
Okay.
Thank you.
Any other questions from the floor? Yeah, I had a small question.
Yes, sir.
Anand, thank you for excellent presentation.
Thank you, sir.
My question is rudimentary.
Sir,
Okay.
no, nothing is rudimentary, sir.
Okay. So we used to talk, traditionally of structure activity relationship. And now we have progressed to in silico modeling. So can AI take it to a step further, predictive modeling of adverse reactions, basically I'm talking, because we are focusing on drug safety issues.
Sir,
So can AI link up all these,
So basically, sir, here when it comes to prediction of ADRs,
Yeah.
when Soibal sir mentioned that it is difficult, that is also right. When if I will say that it is easy, that is also right, because it is the context in which we are looking.
Yeah,
Like what happens is we already know about the ADRs associated with the drugs. So basically the network will start just looking into the words like this patient is taking this drug and he or she is expressing this sort of signs and symptoms. So here it can easily predict the ADRs. But when it comes to newer drug development, we don't have the data of already existing ADRs associated with the drug. But still there are models where like say presence of an alcohol group in this part of the ring can sometimes cause this ADR.
Yes.
But that has not been proven to be very accurate until now. Still research work is going on a lot. Might be in the future that model can come up. So until now, at least as up to as far as I know, as far as up to my knowledge, I have not found any reliable model which can computationally predict the form of structure activity relationship. We can.
Yeah.
Yes. Yes, sir.
Alluding to.
Yes, sir.
Yeah, at least, preempt something, some adverse.
Yeah, it's possible, sir, theoretically, and some models are there. I mean, it's going on in the right direction.
Okay.
Yes,
Thank
sir.
you.
Thank you, Dr. Anand. I have a question on that. Actually, as a clinician, we usually nowadays in this pharma antimicrobial stewardship also in the rampant, we are actually trying to use also and also practice that. In that, the PKPD parameters, because now these data we don't have because now AI is already taken place and automatically with time, definitely it will expand. And what I can see in the foresee as a clinician after within 10 years, maximum thing actually my AI supplement will be there to fine tune my judgment in all aspects.
Agree.
In that one aspect, I utilize PKPD parameters while treating a patient. So because that data I am not keeping now, because without data in the future also I cannot develop a AI model. In the future, like after 2 years, 3 years, we can develop a AI model on that aspect.
So this PKPD or to put it in broad terms, the concept of systems pharmacology, of course, we need to generate data for that. So the hospital has to be equipped with instruments like LCMS and HPLCs and spectrophotometers to measure the drug concentration, along with the facilities for collecting the data with respect to pharmacodynamics also, be it molecular or clinical. So there is no escape with that. We need to generate the data. But if we have data in hand, these days there are a lot of good algorithms present. Even this neural ODE is one of the, it was developed only in 2018. And it has been shown to be much more efficacious than the conventional systems pharmacology approach by using nonlinear mixed effect modeling.
Okay, thank you.
Okay, Dr. Anand. The models for artificial intelligence will need large amounts of data for training.
Yes.
Right. That is the prerequisite. And healthcare data is usually multidimensional data.
Yes.
And that is why AI has a very big role to play.
Yes.
Right. So thank you very much, Dr. Anand. You have made a very complex topic, very lucid and understandable.
Thank you, sir.
Okay. And as I had mentioned, the healthcare data is multidimensional big data, but the advantage is that nowadays we have parallel processing and especially computing with GPUs. Okay.
Yes.
So that makes our task a little bit
Yes.
simpler. By using AI in healthcare, medical professionals can actually, make more informed decisions, which will be grounded on more accurate information because of the, large complex databases. And it will actually save time, cut costs, and it will also result in improving medical records management. Right. So a very nice presentation by both speakers in this session. And I thank both of you for this session.
Thank you,
so
sir.
much.
Please pass the mic.
Sir, what can we do to the data that we feed into the system to make it more structured?
Voice is not clear.
Sir, kindly suggest the ways to make the data that we feed into the system to be more structured, because the clinical data is widely varying.
I'm sorry, what are you telling again? The data structure, what?
Yes, structured versus non-structured data to feed into the AI system.
It's not clear actually. Can you come forward?
Yes.
Structured versus non-structured data and how AI can handle.
Structured versus non-structured data. Okay.
Yes, sir.
So that is why the data scientists are being paid in crores, because they are there to make the data more readable for the computers. Modeling actually takes a less amount of time compared to preparing the data for modeling. AI cannot convert automatically the unstructured data to structured data. AI can handle the structured data well. Even with unstructured data, it will give some output, but that will be pure garbage.
Thank you, sir.
Do we have any more questions? Yeah.
So again, just to put an add-on, so such AI models have not been developed yet. Okay, because AI is always full of surprises. So after a few years, if the model comes where it converts unstructured to structured data, I will not be surprised. But until now, it's not possible. Yeah.
I thank both speakers for such an informative talk, and it was really brainstorming session. I now request our chairpersons to kindly felicitate speakers for this session. I invite Dr. Shoaib Mukherjee sir on the stage. I request Dr. Panda to kindly felicitate I invite President SOPI, Dr. Sandeep Agarwal sir, to kindly felicitate our chairpersons of this session. Thank you, sir. With this, we come to end of day 1 of Society of Pharmacovigilance of India's 20th Annual Conference. Now I request interested delegates to kindly proceed for Ganga Aarti. They may contact Dr. Pramod for the same. Yes, I'll announce that. Now we proceed with the general body meeting of Sopicon. Having GBM in the hall just next to the auditorium. There's a room next to auditorium. We'll be having a general body meeting over there. I request all the delegates to kindly proceed.